Hello, my name is Namshin from Korea Research Institute of Bioscience and Biotechnology. My presentation title is GMStool, GWAS-based marker selection tool for genomic selection from genomic data. Genomic selection is to predict phenotype from genotype information. There has been many approaches to achieve this goal, and what we have found is that if you select optimal markers, we can predict more exactly. And in this approach, we consider epistasis effect SMP to SMP interactions. In previous tool, actually do not consider SMP interactions. In genome, each DNA has SMP interactions if they have significant association with the phenotype. And by heuristic approach, our GMS tool is find the optimal SMP markers from SMP chip to predict phenotype. We used various data from heritability 0.271 to 0.774 in rice and soybean population data. In method section, as we can see, there is a training step, and in machine learning step, and it optimized optimal selection of optimal markers by several methods. In testing step, we can evaluate accuracy by doing this step. As you can see in the B, these markers, we check the addition of new markers by whether the markers improve total accuracy or not. If you iterate over this process by checking accuracy, we can find, and in this step we actually check the SMP interactions from new markers and previous selected markers. Because of that, it actually requires many calculations. And in the B, in figure B, as you can see, there is a very good correlation between genotype and predicted phenotype and those GEBV value. And in this data set, in rice data set, total of number of samples is 13, and number of markers is about 40 SNPs, and we predict DTF, PC, pH, and in summary, you can see the number of samples and mean, median, mean, and standard deviation values. In soybean, there is a 1,928, and the number of markers is about 100,000, and we use the day to flowering time in this example. We first perform genomic association studies to find the correlation and associated markers between genotype and phenotype. As you can see, those heritability values varies between low heritability to high heritability. And this PCA plot shows population structure of these all samples, and those scatter plot between observed and predicted phenotype shows very clear correlation between those observed phenotype and predicted phenotype. In the center, you can see final accuracy for the test set we use the RB, DNN, and CNN method, and actually those DNN and CNN method we have implemented using Python. As you can see, those predicted value using our scheme, and this RRB, RF, DNN, CNN means we can select markers using our scheme. And considering previous studies, this table shows great correlation between genotype and phenotype. For soybean date to flowering time, maximum correlation is 0.864, and for rice pH 0.865, for rice PC 0.770, for rice DTF 0.724. Actually, it is very accurate, and you can find this script in this github.com, Love Moon and GMStool website. Thank you.